All right, thank you so much, Hawa, for that introduction, and thank you so much to the organizers for inviting me to speak today. My name is Rikki Garner. I'm a Helen Hay Whitney postdoctoral fellow in Sean Magison's lab at Harvard Medical School, and I'm currently on the faculty job market. Today, I'm excited to tell you about some of my recent work identifying new and important roles for tissue fluidity in multicellular patterning. Perhaps the most extraordinary and distinctive feature of biological systems is their remarkable ability to self-organize across scales. Millions of macromolecules act in concert to build the cell, and cells in turn range into beautiful and complex structures that enable multicellular life. And yet, life at nearly every scale is stochastic, from macromolecular diffusion and binding, where here I'm showing the diffusion of protein complexes inside of a fission yeast cell, to undulations in cell shape and motility, as exemplified by these dynamic fluctuations in actin density and cell shape in this migrating immune cell, to surprisingly noisy gene expression, cell signaling, and even cell migration during development. A fundamental question at the interface of biology and active soft matter physics is how robust form and function can possibly arise from intrinsically noisy biomolecular and cellular interactions. Addressing this question fundamentally requires quantitative multi-scale approaches, which have presented major challenges both experimentally and theoretically. Throughout my career, I've harnessed my unique background in physics and quantitative cell and developmental biology to identify principled mechanisms encoding biological self-organization and noise suppression from the molecular to the organismal scale. While my PhD work with Julie Theriault focused on the molecular and cellular scales, for my talk today I'd like to tell you about my current work on multicellular patterning. The precise spatial patterning of different cell types within tissues is essential for multicellular life. In this classic example, I'm showing you cell type stripe formation in the developing spinal cord, or neural tube, where different neural progenitor subtypes are nicely arranged into defined layers. One of the major ways that cells form patterns in the embryo is based on gradients of secreted molecules called morphogens, where cells determine which cell type to differentiate into based on the amount of each morphogen that they sense. However, modern time-lapse imaging approaches from our lab and others have revealed that morphogen signaling and gene expression can be exceptionally noisy. And as illustrated by this movie of cell type stripe formation in the zebrafish neural tube, where one cell type here is labeled in green, we know that much of embryonic patterning arises from cellular self-organization, meaning that cells have to actively move and rearrange within the tissue in order to form patterns. And this process of a heterogeneous mixture of cell types sorting into distinct domains is called cell sorting and represents a widespread mechanism of biological self-organization, where here I'm showing just a handful of examples in different species. In order for cells to sort, they have to move. And it turns out that the ability of cells to move and rearrange within a tissue is actually a tightly controlled biomechanical property. This property is often referred to as tissue fluidity, where tissues can transition between a more solid-like jam state, where cells are constrained in place, and a more fluid-like migratory state, where cells are free to move. And tissue fluidity is known to play central roles in development, regeneration, and cancer. Now, there exist many diverse uses and definitions of the term tissue fluidity, so just to be clear, here I'm defining tissue fluidity as the ability of cells to move and rearrange within a tissue. Even though we know that tissue fluidity varies drastically across different embryonic tissues over developmental time, between species, and in healthy and diseased states, we are only beginning to understand how tissue fluidity is biologically controlled and how tissue fluidity impacts the ability of cells to self-organize into patterns. In our recent preprint, we identified tissue fluidity as a critical regulator of cell sorting. Using a tight integration of 3D cell culture to cell sorting experiments, as well as biophysical modeling, we've shown through principles of statistical mechanics and kinetics that tissue fluidity can readily be tuned to freeze, catalyze, or erase tissue patterns, where in a tissue with low fluidity, patterns are frozen because cells can't move. Under high tissue fluidity, cells mix randomly to erase existing patterns, and an intermediate tissue fluidity allows patterning to proceed rapidly and accurately. Overall, our results heavily imply that patterning mechanisms have to be co-regulated and co-evolved with tissue fluidity in order to achieve optimal patterning in different developmental contexts. And these ideas also have really broad and profound implications for how tissue fluidity might control how patterns are formed in development, lost in diseases such as cancer, regained through wound healing and regeneration, and then can be engineered in the creation of synthetic systems. So, having established these exciting new roles for tissue fluidity and multicellular patterning, I'm now determining their consequences for early embryonic development and disease. And for this, I turn to zebrafish development as a model system. The Megasin lab has pioneered in toto imaging and zebrafish, taking advantage of the fact that zebrafish embryos are optically transparent, allowing us to quantitatively visualize and track all of the cells in an embryo over many hours. And these features had the potential to enable me to simultaneously measure tissue fluidity and embryonic patterning in toto at single cell resolution in a living organism. Seeking to develop quantitative assays for tissue fluidity, I first turned to imaging-based lineage tracing, where I injected a high molecular weight dye into one cell at the 16-cell stage and then monitored the spread of the clonal cell lineage over time. Where in a low fluidity tissue, the lineage should remain clustered like an island, and in a high fluidity tissue, the lineage should scatter. Using this approach, I found that the clonal cell lineages, shown here in green, initially remain clustered for the first few hours of development, and then suddenly scatter dramatically across the entire embryoblastoderm within just a few hours. This scattering represents large-scale mixing of cells throughout the embryo. The timing of scattering is actually extremely robust and begins right around 4 hours after fertilization and then continues for many hours throughout epibole and gastrulation. Further, high-resolution imaging of cell shape and actin density, where here only a fraction of the cells in the embryo are labeled, reveal that scattering is driven by large-scale random cell migration, where you can see that cells move with actin-rich leading edges at rapid speeds comparable to immune cells. Paradoxically, this fluidity transition occurs concurrently with the establishment of major patterning events essential for normal development, including morphogen gradients for Wnt, nodal, FGF, and BMP that define the embryo's major developmental axes. At the same time, cell fate mapping starts to become possible right as motility begins. My observation raises the fundamental question: how can robust patterning arise in the context of such extreme active cell mixing? To answer this question, we leveraged a recently published FGF signaling activity reporter from Caroline Hill's lab and injected that alongside a nuclear label to simultaneously track FGF morphogen signaling activity and tissue fluidity embryo-wide at single-cell resolution using a combination of in toto imaging, nuclear segmentation and cell tracking, and FGF reporter quantification. Using this approach, for all of the cells in the embryoblastoderm in 3D, we were able to track how the position, velocity, FGF signaling activity, and nuclear shape features changed over time. Where on the left, I'm coloring each cell's segmented nucleus by its FGF signaling activity, increasing from blue to red, and on the right, I'm coloring each cell by its unique cell identifier based on cell tracking. These in toto measurements have revealed a tight spatiotemporal coordination between motility and FGF signaling, where the onset of motility, plotted here as a function of time in blue, closely followed a sharp increase in the FGF signaling activity, shown in orange. And when we looked at the cell tracks in space, where now I'm coloring each track either by its average FGF activity, shown in the middle plot, or by its average motility, shown on the right, you can see that the spatial FGF activity gradient overlaps closely with a spatial motility gradient, such that cells with high FGF signaling activity appear to move faster. The spatiotemporal coordination between motility and signaling strongly suggests that the embryo is activating cell motility to serve some useful purpose in developmental patterning. And so we're now employing AI and machine learning methods to dig deeper into these massive in toto imaging measurements, where we've already shown, for example, in the rightmost plot, that even shallow neural nets, shown in pink, outperform linear regression models in gray in predicting a cell's FGF activity. And we will further interrogate these models to determine the complex interrelationship between cell movement, nuclear shape and cell cycle, and FGF signaling to ultimately understand how motility and signaling are coordinated to drive robust patterning. In complementary work that I don't have time to discuss today, I've also started uncovering mechanisms underlying the fluidity transition, showing that the transition is initiated by zygotically transcribed factors, largely independent, surprisingly, of morphogen signaling, that drive actin-myosin contractility and cell migration. In addition, we've established perturbations that can be used to inhibit the fluidity transition. And so now we are actively using these perturbations to tissue fluidity to investigate how major developmental axis formation and downstream cell fate specification can be robust in the face of such extreme random cell movements. So in summary, I've developed a rigorous theoretical framework demonstrating that tissue fluidity acts as a critical regulator that can freeze, catalyze, or erase multicellular patterns. And in complementary experimental work, I've identified an early embryonic fluidity transition that actively mixes cells throughout critical patterning events, as well as methods to perturb fluidity in the system, ultimately providing really fertile ground to explore the roles of tissue fluidity in biological self-organization in a living organism. Following up on these discoveries, my future lab will continue combining experiment and theory to investigate multiple mechanisms by which tissue fluidity can contribute to developmental patterning, as well as convey embryonic robustness to challenges such as noise, somatic mutations, and cell death, and determine the molecular and cellular mechanisms that can be used to control tissue fluidity in living tissues, with major implications for IVF, tissue design, wound healing, and cancer. And in this way, we will forge an integrated understanding of how biological systems harness interesting physics to shape and pattern life. Finally, I'd just like to thank my collaborators, my wonderful mentor, Sean Megason, and all the members of the Megason Lab, as well as my funding, and thank you for listening.